So good morning, everybody. I welcome everybody to the NTP Board of Scientific Counselors meeting on July 11th. I am Pam Lyon, the BSc Chair. I'm a professor of neurotoxicology and chair of the Department of Molecular Biosciences at the University of California, Davis. And let's see, I think I'm going to start by asking all of the participants to introduce themselves, and we should have a roster that should be projected right now. There we go. Great. Thank you. So when I call your name, please give a brief introduction of yourself. Eric Bloom.
Good morning, Eric Blum. I'm Vice President of Development Biological Sciences for AbbVie, located in Chicago. And basically, I'm responsible for all non-clinical activities to support drug development.
Thank you very much. Kathleen Gray.
Hello, I'm Kathleen Gray. I'm a research associate professor in the UNC Institute for the Environment and lead the Center for Public Engagement with Science. My expertise is community and stakeholder engagement, particularly around well water contamination.
Thank you. Erin Haynes.
Good morning. My name is Erin Haynes, and I'm the professor and chair of the Department of Epidemiology and Environmental Health in the College of Public Health at the University of Kentucky. I'm a community-engaged environmental health scientist working with communities to help understand their exposure to chemicals. I'm also the deputy director of our NIEHS Environmental Health Core Sciences Center.
Thank you very much. Sean Kimbrough.
Yes, I'm Sean Kimbrough from Morehouse School of Medicine, Department of Microbiology and Biochemistry and Immunology. My expertise is health disparities and community engagement.
Great, thank you. Matt Martin.
Oh yes, Matt Martin. I'm Executive Director and Global Head of Computational Safety Sciences at Pfizer in the Drug Safety Research and Development Department. And my expertise is in computational biology, toxicology, and applications of AI in drug discovery.
Great, thank you. Next slide, please. John Meeker.
Good morning, John Meeker, Professor of Environmental Health Sciences at University of Michigan. My expertise is environmental epidemiology and human exposure assessment.
Great, thank you. Dr. Devin Payne Sturgis is a member of the board, but unfortunately is not able to join us today. So we'll go to Mark Rusi.
I don't think he's joined just yet.
Okay, thank you. Moving on to Veena Singla.
Hello, Veena Singla. I'm a senior scientist with the Natural Resources Defense Council and our People and Communities program, and I work on harmful environmental exposures connected to health disparities and informing policies with the most current science and data.
Thank you. And Janet Yang.
Sorry, everyone, just got a quick phone call. I'm Janet Yang. I'm a professor of communication at University of Buffalo, and my expertise on public risk perceptions and environmental communication.
Thank you very much, and I want to thank again all the board members for joining us today. And so I'd like to move on now and ask a number of other participants in today's meeting who have leadership roles in the various institutes involved with the National Toxicology Program to introduce themselves, starting with Rick Wojcik, who is the director.
Okay, there's Rick Wojcik, the director of NIEHS and the director of the National Toxicology Program. Thanks, Pam.
Thank you. Dr. Robert Sills.
Yes, so I'm Robert Sills, and I'm the acting scientific director of the Division of Translational Toxicology, and welcome everybody.
Thank you. Dr. Brownlow.
Thank you. I am health scientist in the Office of Policy Review and Outreach at the Division of Translational Toxicology at NIEHS, and I'm also the designated federal officer for this meeting.
Thank you. And I realized I skipped Dr. Mary Wolf, which I did not intend to do. Sorry, Mary.
Good day. I'm Mary Wolf. I'm the Director of Office of Policy Review and Outreach in the Division of Translational Toxicology at NIEHS. Welcome.
Thank you. Dr. Gonzalo Gamboa.
Good morning, everyone. My name is Gonzalo Gamboa. I'm a senior science advisor at the Food and Drug Administration, and I serve as the FDA liaison officer to the NTP.
Thank you. Dr. Christina Lawson.
Good morning, everyone. I'm Tina Lawson. I'm the chief of the Field Research Branch and the NIOSH liaison to the NTP. Thank you.
Thank you, everybody. I now invite Dr. Sills to make his remarks regarding his role as interim DTT scientific director. Dr. Sills.
Thank you, Pam. I really appreciate this. Let me make sure that I want to have my screen, my thing, so I could see well here. 1 second. Full screen. Yeah, there we go. That's much better. Yeah. So good morning, everyone, and welcome to our NTP Board of Scientific Council's meeting for the concept review support for predictive toxicology and evidence integration. And as Pam said, my name is Robert Sills, and I am the interim scientific director of the Division of Translational Toxicology. First, on behalf of our director, Dr. Wojciech, and all DTT staff, I would like to thank our distinguished Board of Scientific Counselors and our chair, Pam Lyon, for really taking time out of your busy schedules to provide advice on the concept review. We know that your time is valuable, and we always appreciate your technical and scientific perspectives as we plan for the future. So second, I would also like to thank Kelly Shapkowski and all our DT staff for their hard work in preparing for the BSc contract concept review. In preparing for the reviews, we have multiple practice sessions where DTT staff provide input and contribute to discussions in preparation for a review today, and we really appreciate their collaborations on these activities. Third, we are most grateful to Melanie Brownlow and all DTT staff and our contracting partners who contribute to planning the meeting. As you can see, there are many parts for these meetings to be successful, and it really takes a whole team to get this done. In addition, I also have to thank our Office of Acquisition for their important partnerships. So this is my first BSc meeting as the interim DTT scientific director, and I would just tell you a little bit about what I've been doing since I started this role. So Rick said to me as I started this role, he wanted to make sure that DTT is sound and stays really productive, and we really want to be in a place where we prepare the organization for the new scientific director. So I have two goals when I started this position. The first goal was to engage with staff and support everyone. We've had many changes in DTT over the past 5 years, starting from management changes. We've had a name change from the Division of National Toxicology Program to Division of Translational Toxicology, and other changes that have positioned the organization to be really forward-thinking. And so my role was to really support staff during this time. And so for goal one, which is again, engaging staff and supporting everyone, that's the first one. And the second goal is to build on our scientific strengths. But for goal one, the first thing I wanted to do was really initiate listening sessions among all the branches, offices, and individual staff. And this has been really fantastic. So I started, before I became the acting scientific director, I was the chief of the pathology branch, and in that role, I was served as a branch chief. I thought it was important when I took on this role as the acting scientific director was for me to really get to know staff at this level. So I've been meeting with all the staff among the branches, offices, individual staff, really to learn more about their scientific innovations and about how I could really support them during this transition. So some of the things we've also been doing is really seeking their input on workplace flexibility. How is it working? How is work-life balance working for you? And again, we've had some really important changes regarding increasing efficiency in our administrative structure. So another goal was to continue to build successful partnership between our staff and our administrative team, and then continue to provide leadership opportunities in the areas of career development, and most importantly, to be an advocate on our management team, along with our supervisors for all staff. And then finally, in regards to goal one, is really represent all staff on our senior leadership committee that is led by our director, Dr. Wojciech, and on the NIH Scientific Directors Committee. We have meetings twice a month in Bethesda. In regards to my second goal, it's to build on the scientific strengths of the DTT. And so I'm pleased to say that we have excellent research capabilities and R&D contracts in place to support DTT research. And that's the reason we're having this meeting today for our board of scientific councils to provide advice so that we have the best capabilities, the best, we're making sure that we have the best innovations to get our science done. And again, we appreciate your participation in this today, because without the correct research capabilities, innovations, we wouldn't be able to do the work we're doing today. And in addition, we have a number of ongoing program management team scientific updates from our 4 areas, strategic program areas, including our health effects innovation programs, our exposure-based research, our responsive research, and strengthening capabilities research. Another goal is to foster, continue to foster collaborations across NIHS between DTT, DIR, and DIRT, and our NIH ICs, other ICs. So I have some good news for you today. So starting tomorrow, we will be interviewing 3 outstanding candidates for the permanent DTT scientific director position. So these candidates are, the first one is Christina Thier, and she is a director of the Chemical and Pollution Assessment Division, Center for Public Health and Environmental Assessments at EPA. So she'll be interviewing on Wednesday and Thursday of this week. And then our second candidate is Russell Thomas, and he's the director for Center for Computational Toxicology and Exposure at the EPA, and he'll be interviewing next Monday and Tuesday. And then our third candidate is Heather Pattisall, and she is an Associate Dean for Research, College of Biological Sciences, North Carolina State University, and she'll be interviewing next Wednesday and Thursday. So thanks again for your important advice in this review process, and I welcome any questions or comments that you may have.
Thank you very much, Dr. Sills. Are there any questions or comments from the BSC?
And can I just chime in here that
Sure.
Robert's doing a fabulous job. So I just want to personally thank him for his willingness to step in this very important role during this interim period.
Thank you. Thank you to both of you. And congratulations on a great roster of candidates for the position. I'm very excited. That's wonderful. Okay, if there are no questions from the BSC, we'll move on then to see conflict of interest and housekeeping issues that will be described to us by Dr. Brownlow. Dr. Brownlow.
Thank you. So I will proceed to reading the conflict of interest statement and just going over some of the housekeeping items, as Dr. Lyman mentioned. So the members of the NTP Board of Scientific Counselors serve as individual scientists and not as representatives of any organization. Each member is to exercise judgment prior to or during the meeting as to whether a potential conflict of interest might exist relative to the topic under discussion due to his or her occupational affiliation, professional activity, or financial interests, or those of their spouse, minor child, partner, or organization. Board members are asked to bring to the attention of the committee and of the federal officer any potential conflict of interest that might pertain to the topic being considered at the meeting. Should there be a potential conflict of interest, it will be handled in accordance with departmental policies and regulatory requirements. Thank you. And for housekeeping, so this is a public meeting and it is being broadcast to a remote audience. So when speaking, please remember to introduce yourselves to the listening audience. We also ask that since this is a public meeting, please reserve the use of the Zoom chat box to technical issues only. If you're having any technical issues, please notify me or anybody from our AV team. All discussions in the meeting will take place in the public forum and will be captured in the Meet meeting minutes. We also ask presenters and speakers to mute yourselves and turn off your video unless you are speaking as part of Zoom etiquette. Slides and video recording will be made available on the meeting webpage as soon as they meet the government requirements for accessibility. And lastly, minutes will be posted to the meeting webpage once they have been reviewed for accuracy by the board members, approved by the chair, and also meet government requirements for accessibility. Thank you, everybody.
Thank you. So I have just received a note that Dr. Mark Russi, a BSc member, has just joined us. Dr. Russi, would you please quickly introduce yourself?
Apologies, yes. Hi, I'm Dr. Mark Russi. I'm a professor of medicine and epidemiology at Yale University, and the area of expertise is occupational medicine.
Great, thank you, and I'm happy you're able to join us this morning. So now we'll move on to a presentation by Ms. Kitzmiller from the NIEHS Office of Acquisitions, who will give us our instructions for the concept review. Ms. Kitzmiller.
Hello, good morning, everyone. My name is Erica Kitzmiller, and I am a contracting officer here at NIEHS. I want to echo Dr. Sills and begin by thanking each of you for dedicating your valuable time to be here and serve as a member of the Board of Scientific Counselors. I want to emphasize that the work you do here is critical in the review and evaluation of our research programs, and that includes the concept review meeting being performed today that is required for our research and development contracts. 42 CFR Part 52 and NIH Policy Issuance 6315 TAC 1 requires that a peer review group or advisory council must first review the project concept before my office can release a contract solicitation involving biomedical research and development. As part of this concept review, we will have a high-level discussion of the project to identify the basic purpose, scope, and overall objectives of the work. You are reminded that we will not discuss specifics, such as the technical approaches, protocols, or product specifications, and we will not include details of the acquisition approach. This will help preserve the integrity of our procurement process and allows us to maintain an open meeting. As you review the contract concept provided today, please consider the overall value and scientific relevance of this project and whether or not it supports our goal of protecting public health. I also ask that you consider the following 5 items. Number 1, the significance of the goals of the proposed research activity. 2, the availability of technology and other resources necessary to achieve these goals. 3, the extent to which there are practical, scientific, or clinical uses for the expected results. 4, the adequacy of the proposed methodology. And 5, the appropriateness of the use of a contract mechanism to support the activity. With that being said, I am now happy to turn it over to the contracting officer's representative, Dr. Kelly Shipkowski, to discuss the projects.
Sorry, my mouse is being wacky this morning. Before we do that, I just want to ask if the BSc members have any questions for Ms. Kitzmiller. If you do, please use the Zoom function to raise your hand. And when I call on you, please state your name. Are there any questions from the BSc? Okay, seeing none, I am going to turn it over now to Dr. Shipkowski and ask her to make her presentation. Thank you.
Thank you, Dr. Lyon. Okay, are you able to see my slides?
Yes.
Perfect. So good morning, everyone. My name is Kelly Shipkowski, and I'm a health science evaluator in the Office of Program Operations here at the Division of Translational Toxicology. I'm also a contracting officer's representative, and today I'll be presenting a contract concept for review on predictive toxicology and evidence integration support. So here's a brief agenda of what I'll be going over this morning. I'll start off with a high-level introduction to contract concepts and DTT contract support in general, and then move into the actual concept review itself. So starting with the introduction, why are we here today? As Erica just mentioned, all of our research and development or R&D contracts require external peer review of the corresponding contract concept. And what that review essentially is it's a review of the use of a contract mechanism to secure the needed capabilities that we're looking for. And this requirement applies to contracts that are new. It applies to contracts that are recompetitions with changes in their statements of work. And it also applies to contracts that have been ongoing for 5 or more years since the last review. And the concept that we'll be discussing today falls in these bottom 2 buckets. This review is required prior to requesting any kind of proposals, and it's important to emphasize that a single contract concept may encompass multiple statements of work, solicitations, and/or contracts. And the resulting contracts will be with NIEHS, but it's important to note that the work performed under these contracts may also support NTP projects. So when thinking about DTT contract support, I think it's helpful to have at least a high-level understanding of how our division's funding is allocated. So DTT's funding we have 2 buckets of funding. There are intramural or IR funds, and these are used for things like salaries and benefits, operating expenses, and travel. And then we also have our research and development or R&D funds, which are used for contracts and interagency agreements. And as you can see with this pie chart here, over 3/4 of our budget is comprised of R&D funds, which significantly influences the extent to which we utilize contract support to complete our work. So moving on, I think it's also important to talk about how DTT contracts are managed relative to our available funding. And so, as I mentioned at the beginning of my presentation, I'm a contracting officer's representative, or a COR, and CORs are authorized to perform specific technical and administrative functions under a contract. There are 3 levels of COR certification, and certification requires a combination of experience. The highest level of certification, the COR 3, requires a minimum of 2 years experience on federal projects. There are also a number of formal training courses that we're required to take. There's also a very detailed transcript that we are required to prepare that outlines our experience with a number of different competencies. And then ultimately, certification requires approval by both NIH and HHS. Most, but not all, of the CORs within DTT reside in the Office of Program Operations, or OPO. And what our office does is we provide centralized oversight of DTT's budget, as well as contract acquisition and management. And it's important to note that all of the staff in OPO also provide a wealth of scientific and project management expertise. DTT contracts support a number of different areas across our division. And while the bulleted list that I have here is by no means comprehensive, my goal was really to demonstrate the wide variety of different capabilities across our division that utilize contract support. And today, as you're aware, we will be focusing on support for predictive toxicology and evidence integration. And I also want to highlight that for those of you who were BSc members with us last year, today's meeting is similar to the concept review that was conducted in December, where we reviewed contract concepts for chemistry, pathology, and toxicology support. Next, I'd like to talk a little bit about how these different capabilities that we use fit into our division's workflows. And what I am showing you here is our translational toxicology pipeline. And for those of you who may not be aware or familiar with our pipeline, this was introduced in early 2018 by our former scientific director, Dr. Brian Berridge, as part of a larger effort to strategically realign DTT's portfolio. And what this image represents is a pipeline of capabilities that can be developed and utilized to accelerate our progress toward becoming a more predictive, precise, and preventative science, and ultimately help us provide data that are translatable, predictive, and timely. Within this pipeline, predictive toxicology and evidence integration support contracts represent a variety of DTT capabilities, which I've tried to highlight here. It's important to recognize that there is a lot of interconnectivity between these capabilities and other DTT contracts. And I think it's helpful to note that the relationships among these different capabilities are such that the outputs of one contract may become inputs for another. I'd also like to highlight some other mechanisms that we have within NIEHS for collaboration and support. First off, there's the NIEHS Office of Technology Transfer, and here I have some examples of some of the different agreements that are coordinated through that office. The government is not able to add funding to these agreements. However, DTT does utilize these for some of our collaborations. There's also the NIEHS Division of Extramural Research and Training, and this division functions primarily via grants and cooperative agreements. And these agreements do require grant authority and funding, which DTT does not have. However, DTT scientists may serve as subject matter experts on some of these cooperative agreements. So moving on to the concept review, I'd like to start off with just a brief history and overview of the requirement that we'll be discussing today. So support for predictive toxicology and evidence integration is essential for DTT's research program activities. The scope and the breadth of the required support exceeds the internal staff resources that are available at NIEHS, and support for these activities has been obtained via contract resources for over 40 years. And the type of support required has evolved over the years to align with changing division needs. And we continually anticipate further alignment as we update our DTT strategic portfolio. And so bringing us back to this figure, I do just want to reemphasize where along the pipeline predictive toxicology and evidence integration support fit in when looking at the different DTT capabilities. And for this concept, I'm going to use the next few slides to talk about 9 specific key capability areas for the concept that we're reviewing today. And the first 3 key capability areas are developing, evaluating, and implementing computational tools and methodologies, developing, validating, and implementing alternative approaches for toxicity testing, and then searching, screening, evaluating, and synthesizing information from environmental health literature. The next 3 key capability areas include compiling and preparing reports for publication, editing documents, and providing technical review for completeness, accuracy, and readability, managing logistics for external peer reviews, including identifying and maintaining a directory of scientific experts, conducting conflict of interest screening, and preparing documents for peer review, and then managing logistics for remote and in-person events, including Federal Advisory Committee Act or FACA meetings, interagency engagements, and public workshops. And then our last 3 key capability areas are assessing the quality, so the accuracy, consistency, and completeness of products, including toxicology study reports, literature evaluation reports, manuscripts, and data pipelines by comparing their content to original records and documentation, identifying knowledge gaps and formulating discrete research questions, and then lastly, tracking contract project spending, milestones, and deliverables, and managing personnel with efficient coordination of schedules to meet project needs. So before we move into discussions of the contract concept, I do want to reiterate some of what you heard from Mrs. Kitzmiller before my presentation. So because this meeting is open to the public, the discussions are limited to a review of the general project purposes, scopes, goals, and various optional approaches to pursue the overall objectives. The meeting will become closed to the public if the discussions turn into the development or selection of details for the projects or any requests for proposals. And some of these examples of details include things like specific technical approaches, protocols, statements of work, data formats, and any specifications. And the overall intent of this is to protect the free exchange of the board group members' opinions while avoiding any premature release of details on proposed contract projects or RFPs. And so here's the charge to the Board of Scientific Counselors today. NIEHS proposes to obtain support for these activities via contract mechanism due to the scope of the required capabilities and the availability of personnel with relevant experience to perform these activities exceeding the internal resources available. Consideration should be given to the significance of the goals of the proposed research activity, the availability of the technology and other resources necessary to achieve those goals, the extent to which there are practical, scientific, or clinical uses for the expected results, and the adequacy of the proposed methodology. And so ultimately today, the BSC members are being asked to review the contract concept and vote on whether a contract mechanism is the appropriate mechanism to support the proposed activities. And with that, I will take any questions or comments. Thank you.
Thank you very much, Dr. Shipkowski. So I will open this up to the BSC for any questions. Again, please use your raise hand function, and I will call on you and introduce yourself when I do. Eric Bloom.
Yes, Eric Bloom here. Kelly, I've got a question actually in the scope. You're going to generate a high volume of data, and I was wondering, is required infrastructure, the data storage capabilities, etc., is that included in the scope, or is that completely separate?
So I think a lot of the details of the scope are outside of the scope of this meeting today, but in general, a lot of the data storage requirements that we are supported by other contracts within that our division has.
Okay.
The majority of the data, we as the government receive all of the data from any of these projects, and then how that data is managed is ultimately supported by some of our other contracts.
Okay, thank you.
Great question. I had the same question. Matt Martin.
Yeah, maybe similarly, but kind of more in the compute space. So especially with, if you're working in the extraction of information from literature with large language models and maybe needing to extend them and things like the compute needs, not just basic infrastructure like storage, but access to GPUs and things like that are getting more prevalent. So just wondering if not so much in scope or out scope, but well, maybe that is part of it, but also whether you're thinking about that in terms of making sure those capabilities are built into, and have access for either your contractors or as a general service to all of the NTP.
Yes, that is certainly considered when we're thinking about these projects, especially since, NIH and above has an assortment of different guidelines related to, data sharing and storage that we all adhere to. So that's certainly something we take into account when putting these together.
Thank you, Matt. Any other questions from the BSC? For clarification, is everyone on the BSC clear on what our charge is and the specific parameters of what we're to weigh in on? Okay, it sounds like this is a pretty straightforward request that you're asking of us, and the board is confident in what they need to do. So we do have a public comment. There was one written public comment that was received. This is available to the public on the meeting webpage and was provided to the board members in advance of the meeting. This was submitted by Randolph Ashton on behalf of NeuroZeta LLC. And there is a URL down at the bottom of this page if anybody's interested in reviewing that. In addition to this one written public comment, there was a request to present an oral public comment by Dr. Ashton. And so he will be presenting on behalf of NeuroZeta and will join our Zoom meeting, has 5 minutes. Dr. Ashton, once you get your presentation uploaded, please introduce yourself and you have 5 minutes for your presentation.
I just joined.
Okay, great. Thank you. Please upload your slides and introduce yourself.
Okay. Are you seeing my slide?
Yes, we are. Thank you.
Okay, great. Are you ready for me to go now?
Yes, please. You have 5 minutes. Thank you.
Great. So as an example of maybe potential contracting, so my name is Randolph Ashton. I am a associate professor of biomedical engineering at the University of Wisconsin-Madison, and also acting CEO of NeuroZeta. This technology that I'm presenting to you, our RoseZeta Array platform, was initially funded by the EPA, part of a STAR grant, and now the translation of this has been funded actually by NIEHS. So our platform is for developmental neurotoxicity screening, and essentially we think that human brain and spinal cord development is a critical facet of human development that needs to be protected. And essentially, we can't test on models of actual human embryos. And so a powerful technology known as organoid technology has been developed over the past decade or so. This basically allows us to recapitulate facets of CNS morphogenesis or development in a dish. But this technology, of course, has limitations in its scalability and reproducibility of the tissue structures that are formed, and then analysis of these structures. And so our solution to this is to bioengineer this in vitro neuroorganoid morphogenesis. And essentially, we recapitulate the earliest stages of brain and spinal cord development or formation of this neural tube structure, which is essentially a polarized tube of neural epithelial cells and neural stem cells. When you differentiate your pluripotent stem cells to these tissues in vitro, they spontaneously form these type of structures, which we call neurorosettes. But they do this in a spontaneous and uncontrolled fashion. And basically, we have created culture substrates that provide geometric confinement in order to standardize the formation of these single neurorosette structures, providing spatial and temporal control over the formation of these three-dimensional tissues, which have the polarized structure of the neurorosettes at the center of the tissue. Go forward. Yeah. We combine that with other protocols for generating not just forebrain tissues, which most organoids our platforms create, but also we can generate tissues of the hindbrain through the spinal cord, different aspects. And then we use computer vision in order to create an AI-based high-throughput imaging analysis platform for analyzing the tissues that we can analyze, images we can collect via confocal microscopy. And this gives us our RoseZeta Array platform, where we essentially can model the formation of different portions of this neural tube along the rostrocaudal axis of the brain and spinal cord across a single well plate and conduct high throughput screening. We've standardized this for forebrain, cervical, and lumbosacral regions, and this essentially is a 5 to 7 day culture period, where at the end of that culture period, we have a single analysis step that gives us the information on cell viability, proliferation, neural differentiation, and rosette formation. The key here is that we are recapitulating morphogenesis, and morphogenesis happens in a particular space and time, and we've standardized that so we can detect sensitive perturbations to that process. In practical application, you basically take your cryopreserved cells, you can see them directly onto our engineered microwell plate. You can grow the tissues in the presence or absence of various chemicals. At the end of that culture period, you have hundreds of tissues per well that you can go through and collect images from, run that through our image analysis software, and you can essentially spit out the data to give you nice dose response curves for various chemical exposures. You can also play with genetics based on the cell type that you use to make your RoseZeta Array. And this starts to give us not just whether something is toxic, but allows us to have more mechanistic insight because of the juxtaposition of these curves. Different pathways control different facets of the different curves, and so we can start to understand which pathways are being more affected versus others. Our platform essentially takes the standard in vitro DNT screening battery, streamlines that, replacing a handful of those assays with our single assay. And then we're also working on second generation version of this to cover additional aspects of human brain and spinal cord development to essentially have sort of a 2-assay battery that would cover a lot of the current DNT screening battery.
You have 1 minute to wrap up. Sorry, 1 minute to wrap up.
What we've done so far is to show that this can work with DMSO solvated chemical libraries. We have excellent reproducibility across the different regions in our assay. We can integrate human metabolism into our screening. We've done this for a handful of compounds. We're doing more with the STTR grant, which gives us really good sensitivity and specificity. And we do note the importance for using forebrain versus spinal rosette arrays. So we hope that the National Toxicology Program would be interested in this assay, particularly to use for contract human D&T screening. We also can provide the well plates and kits if they want to do this in-house. And we have a web accessible image analysis software that will be coming on board soon. And we are in the middle of our fast track STTR from NIHS to screen 100 compound library for further data. And with that, I can take any questions.
Thank you very much. Would you mind turning off your presentation so we can see all the members? Thank you. Are there any questions from the BSC for Dr. Ashton? And thank you for a nice presentation, Dr. Ashton. Questions from the BSC? Any questions from the NIEHS NTP staff? Okay, hearing none. Again, we thank you for a really nice presentation, very exciting technology. And I believe that we will turn to our BSC discussion. And so Dr. Brownlow, please advise here, but I believe Dr. Ashton has to leave our meeting now. Is that correct?
Yes, that is correct. Thank you very much, Dr. Ashton.
Thank you.
Thank you. Okay. So we are now to the point of our meeting for the BSC discussion and action. So just to remind everybody, our role here today is to vote on whether a contract mechanism is the appropriate mechanism to support the proposed activities that Dr. Szybkowski reviewed for us. So I'll open this up to ask if any of the BSC members have any questions or comments they'd like to start the discussion. Again, if you do, please raise your hand and I will call on you and introduce yourself. Okay, so I guess the only comment I would have as the chair of the BSC is that this seems like a very straightforward activity that we're being asked to consider, that there is, as it was noted earlier, a great deal of support needed to really follow through on the various activities that were described to us, and that to me, at least, the contract mechanism seems appropriate. It's been used successfully in the past. And again, this is to determine whether this mechanism is still appropriate. And these are not new activities that are being proposed, but a 5-year review and or continuation of activities that have gone on in the past. A lot of it does seem to be logistics support, sort of data management, review of contract mechanisms, et cetera. Kelly, do you want to add to that at all? Is there anything I missed there that you would like to be sure we consider?
No, I don't think so. I think you captured it. Thank you.
Okay, thank you. All right, any additional comments, concerns from the BSC? I think this should be straightforward then. So if there are no questions or comments, I propose that we can go basically, I need a motion, actually, I guess, to whether the contract mechanism is the appropriate mechanism to support the proposed activities. Does anybody make the motion?
Sorry, I was just
Go
going to
ahead.
say, Mary seems to be saying something, but she's muted.
Oh, sorry.
So Melanie, I wasn't sure if you were able to show the voting sheet or not so that we could get that captured before we have the motion. Thank you.
I am, thank you. I had some technical difficulties, but I opened it back up. So thanks. Yes.
So just to clue in the BSC, we will go through and call your name out to vote on this. If you vote either no or abstain, you have to give a reason for that. So, but before I do, I need a motion from one of the board members as to whether or not the contract mechanism is the appropriate mechanism to support the proposed activities. Martin.
Yeah, I'm happy to make the motion to vote yes, that the contract mechanism is the appropriate mechanism to support the proposed activities.
Thank you. Matt Martin made the motion to support the proposed activities via the contract mechanism. I need someone on the BSC to second that. Erin Haynes.
Yep, I'd be glad to second that motion.
Seconded by Erin Haynes. Okay, now we are going to vote by verbal acknowledgement. So starting with Eric Bloom.
Yes.
Kathleen Gray.
Yes.
I'm sorry, could you repeat that, Kathleen?
Yes.
Thank you. Erin Haynes.
Yes.
Thank you. Sean Kimbrough.
Yes.
Matthew Martin.
Yes.
John Meeker.
Yes.
Mark Rusi.
Yes.
Vina Singla.
Yes.
And Janet Yang.
Yes.
So it appears there's unanimous support for the contract mechanism to support these activities. So I think we are concluded. So I have no additional closing remarks other than to thank the BSC members for their engagement today and to thank the NTP staff for putting together a really clear presentation and a really clear charge to us, which made our decisions relatively straightforward today. I'll ask Dr. Sills if he has any closing remarks for us.
I just want to echo what you said, Pam. Thank the Board of Scientific Counselors for really doing an outstanding job of reviewing and providing comments. And then I'll turn it over to Rick for any comments he may have.
No additional comments other than to thank everyone. And Pam, great job at the helm here for the BSC. So a very efficient meeting, and thanks everyone for your engagement.
Thank you very much, both Dr. Sills and Dr. Wojcik. Dr. Brownlow, any closing comments from you?
Thank you. I just want to thank everybody for preparation for the meeting. Thank you to all who have worked to put the meeting together. And just a reminder that slides, the video from today will be made available on the meeting webpage as soon as they meet government requirements. And likewise, meeting minutes will also be available once they have been reviewed by the board and approved by the chair. Thank you.
Thank you very much, Mylene. And thank you to everybody. This meeting is now adjourned.
Thank you.
Thank you.
Bye.
Thank you.
Thank you.
Thank you. Bye-bye.